Hey, welcome back. So let's jump right into one of the biggest ideas in biology, the central dogma. Now, we usually see it as this super simple straight line, but today we're going to peel back the layers and see that this simple path is actually built on some of the most amazing solutions to some really huge engineering problems. So here it is, that classic diagram you probably remember, right? DNA makes RNA, RNA makes protein. It looks so neat and tidy. But believe me, this elegance hides an entire world of microscopic machinery that works with a kind of precision that is just, well, it's absolutely mind-blowing. So, we're going to approach this a little differently today. We're not just going to memorize a diagram. We're going to solve a series of puzzles. We're going to unpack 4 of the most ingenious problems that your cells have to solve every single moment of every single day, just to keep you going. Ready? Let's do it. Our very first puzzle starts at the most fundamental level, making a copy of DNA. And the challenge comes from the structure of the DNA double helix itself. It's a famous little headache known as the lagging strand problem. Okay, so here's the deal. The 2 strands of DNA are what we call anti-parallel. Think of it like a highway, but the 2 lanes are going in opposite directions. The problem is the main enzyme that builds new DNA, DNA polymerase, is strictly a one-way vehicle. It can only travel in one direction, from 5' to 3'. So how on earth do you copy both sides of the road at once if your construction crew can only move in one direction? The cell's solution is just brilliant. It doesn't even try to copy them the same way. For one strand, the leading strand, the polymerase just gets on the highway and cruises along in one nice, smooth, continuous piece. But for the other one, the lagging strand, it has to do this kind of awkward start and stop maneuver, building it in little pieces called Okazaki fragments. It really is like a tiny, super coordinated construction project. First up, an enzyme called primase comes in and lays down a little RNA primer, basically saying, start here. Then DNA polymerase jumps on and builds a short stretch, backwards. This whole thing repeats over and over. Then a cleanup crew, an exonuclease, removes those starting primers, polymerase fills in the gaps, and finally, an enzyme called DNA ligase comes in like molecular superglue and seals all the fragments together. It's this incredible rhythmic dance. Okay, puzzle number 1 solved. The DNA is all copied. So now for puzzle 2. How do you read the instructions? The cell needs to make an RNA copy of a gene that's called transcription. But where exactly does it start? This is a classic needle in a haystack situation. You know, the genome is gigantic. We're talking millions, sometimes billions of letters long. And the enzyme, RNA polymerase, has to find the exact starting line for one single gene among thousands. If it's off by even one letter, the whole protein it's trying to make could be useless. So how does it find its spot? Well, it turns out life has come up with 2 main strategies here. In bacteria, the system is really simple and direct. But in eukaryotes, that's organisms like us, it's way more complicated. It's almost like you have to build an entire launch pad before the rocket, the polymerase, can even land. You gotta love the bacterial system for its sheer efficiency. The RNA polymerase has a little helper protein called a sigma factor. And this sigma factor is basically a guide. It just scans the DNA looking for 2 specific sequences, the -10 and -35 boxes. When it finds them, it locks on, and boom, it puts the polymerase right at the starting line. Simple, fast, direct. Eukaryotes, though, they build that launch pad. It all starts with a sequence called the TATA box. A whole team of proteins called transcription factors have to show up and bind to the DNA first. They assemble one by one, like stacking blocks, creating this big protein structure. And only when that whole initiation complex is perfectly built can the RNA polymerase finally land and get to work. It's a much more controlled, deliberate process. All right, let's move on to puzzle number 3. We've made our RNA message. Now the cell has to actually translate that message into a protein. And this all goes down inside the ribosome, which is basically the cell's protein factory. The challenge now is all about mechanical precision. The ribosome has to read the messenger RNA blueprint 3 letters at a time, grab the exact right amino acid for that 3-letter code, and then stitch them all together in the perfect order. It's a high-speed assembly line that has absolutely 0 room for error. So, first things first, how does the ribosome even know where to start reading the blueprint? Well, in bacteria, there's another brilliant little recognition trick. The mRNA has a special sequence called the Shine-Dalgarno sequence.
and this sequence physically matches up and binds with the RNA inside the ribosome itself, literally locking the blueprint into the perfect starting position. It's a perfect physical alignment.
Once it's locked in, the real work begins across 3 little workstations inside the ribosome. You've got the A site for arrival, where the new tRNA carrying an amino acid shows up. The P site for polypeptide, where the growing protein chain is held, and the E site for exit, where the empty tRNA gets kicked out. These 3 sites are the whole key to the ribosome's clockwork cycle.
And that cycle is just stunningly precise. It goes like this. 1. A new tRNA arrives at the A site. 2. The protein chain gets handed off from the P site to the new amino acid at the A site. 3. The entire ribosome physically slides over exactly 3 letters on the mRNA. This shuffles all the tRNAs to the next spot, and the now empty one gets ejected from the E site. And then the cycle just repeats, over and over, adding amino acids with incredible speed and accuracy. Okay, that brings us to our 4th and final puzzle, which might be the most dynamic one of all. It's something pretty much unique to eukaryotes, and it's called splicing. This is where the cell basically becomes a master film editor for its own genetic script. So in eukaryotes, when you first make an RNA copy of a gene, it's a rough draft. It's got the parts you need, called exons, but they're broken up by these long non-coding stretches called introns. You can think of it as junk DNA. The cell has to snip out all the junk and stitch the good parts together perfectly. One mistake and the whole message is garbled. How does it pull that off? This is what that rough draft or pre-mRNA looks like. The blue parts are the exons. Those are the keepers, the bits that will actually become the protein. And that orange section in the middle is the intron. That's the part that has to go. This incredible editing job is done by a massive machine called the spliceosome. What it does is amazing. It grabs one end of the intron, bends it over into a loop, and actually uses that end to attack another spot within the intron itself. It's this wild molecular gymnastics move that forms a loop and isolates the intron from the good stuff. And that loop has a name. It's called a lariat. And for good reason, it looks exactly like a cowboy's lasso. And forming this lariat shape is the absolute key to snipping out the intron cleanly. And here it is, the final cut. The spliceosome snips the lariat completely free, it'll just get broken down and recycled, and in that exact same moment, it perfectly stitches the 2 exons together. What you're left with is a clean, mature mRNA, now a continuous message, all ready to head to the ribosome. So from the clever backstitching on the lagging strand, to building a launch pad for transcription, to the clockwork precision of the ribosome and the incredible lasso of the spliceosome, these are not just static diagrams in a textbook. They are dynamic, elegant solutions to real engineering problems. And they are all happening trillions of times a second inside of you, right now. It just goes to show you that behind those simple arrows of the Central Dogma, there is an entire universe of mechanical wonder. And it really makes you think, what other incredible puzzles are these tiny, exquisite machines solving at this very moment that we haven't even discovered yet? Thanks for joining me.